So good morning students. Today we will discuss about some more structural details of prokaryotic cells. Okay. In the last class we had talked about the plasma membrane. Now next we will talk about the cell wall. Okay. See, next one. Next that is the cell wall. Here, the cell wall of prokaryotic cells, where first the innermost membrane which is enclosing to the cytoplasm, that is the plasma membrane. And that plasma membrane, here you see, this is the plasma membrane. It is covered by a rigid envelope. It is covered by a strong and rigid cell that renders mechanical protection and provides the bacteria their characteristic shape. Okay, here there is a rigid structure, and that rigid structure is the cell wall. What is the role? What is the function of a cell wall means? So here, cell wall of bacteria. First of all, it provides the mechanical protection. What it will do? It provides the mechanical protection. The second thing, here it will provide a particular shape. It gives a shape to a cell. Okay. The next thing here, the cell wall. And one more thing, very important thing, all the prokaryotic cell, all the bacteria possesses a cell wall except one bacteria, one unicellular organism or one prokaryotic cell, which is in which there is an absence of what cell wall. What is that organism? Is that it is a mycoplasma. Mycoplasma is one of the prokaryotic cell in which the cell wall is absent. Which the cell wall is absent. The next thing here, that cell wall of bacteria differs chemically from the cell wall of plants in that contains protein, lipids and carbohydrates. Okay, so it may also contain chitin but rarely any cellulose. Here the cell wall of prokaryotic cell is different from the plant cell wall. What is the chemical composition of the cell wall over here? Means chemically, it is cell wall is made up of proteins, lipids, carbohydrates, and some of the cell wall of prokaryotes will have the cellulose. Only some, not all, but in all, which are that particular chemical components means that is the protein, lipids, and carbohydrates. Okay, so the next we will go for the, after cell wall we will go for the capsule. Next structure, that is the capsule. So that we will see here. Capsule is there. In some bacteria, the cell wall is surrounded by an additional slime or a jelly-like substance. To that we can call it as the capsule. So here, next one, next character, that is the capsule. Here, in some prokaryotic cells, first of all, the first membrane was the plasma membrane. After that, this plasma membrane is covered by a hard, rigid or tough, rigid structure, that is the cell wall. And the cell wall here, it is made up of protein, lipids and some of the polysaccharides. Among that polysaccharides, if you talk means that is the cellulose. Okay. The next here, in some of the prokaryotic cell, it is enveloped. This cell wall is enveloped by a thin slime or a jelly-like layer, and this jelly-like layer is known as the capsule. Okay, see here, in some bacteria, only in some bacteria you will find capsule, not in all prokaryotic itself you will find the capsule. Okay, so here it is this capsule is made up of a gummy substance or a jelly-like substance, to which we can call it as the gummy substance. So here that gummy substance, what it is means, it is made up of what? Mucilaginous layer. Okay, mucin-like substance will be present, which gives a gummy-like substance or jelly-like substance to the capsule. Okay, then it is secreted. From where it is going to secrete this mucilaginous layer? Means this layer is secreted from the plasma membrane. Secreted from the plasma membrane. From this layer is going to be secreted. What is that? That is the capsule. So what is the function here means, first thing, what is the function of capsule? It acts as a protective layer. Okay, it's one of the protective layer, or it serves a protection to the particular bacteria against what? Means it serves as a protective layer against the phagocytes. Against the phagocytes and by viruses. Certain cells which are damaging the bacteria or the prokaryotic cell, this particular capsule layer is going to protect that particular what we call bacterial cell from the phagocyte and by some viruses. The next thing here, it also helps in regulating the concentration of what? Of ions and molecules. It also helps in regulating the concentration of ions and some other molecules. So this is about the capsule. First plasma membrane, second cell wall, and third is the capsule. Next we will go for the next cell organelle over here, that it is the cytoplasm. Next one, cytoplasm. Here, whatever the plasma membrane which is enclosing a matrix-like substance, this is here, it is a matrix-like substance. Okay, that here, this is the plasma membrane which is enclosing the matrix substance. Okay, this consists of matrix, or else we can also call it as the cytosol. What it is, we can also call it as the cytosol. Okay, so here this is the ground substance of the cell, or where we can also call it as the seat of all metabolic activities. Cytoplasm is also known as the seat of all metabolic activities. Okay, because all the chemical reaction, all the cellular activities or the metabolic activities will take place in this ground substance, that is the matrix of the cytoplasm. Okay, so the cytosol consists of water, protein, lipid, carbohydrate. It consists of water, protein, lipid, carbohydrates, then it also contains different types of RNA molecules, and various smaller molecules, Okay, this is the chemical composition of what? Of cytoplasm, where it consists of water, protein, lipids, carbohydrates, RNA molecules, and various smaller molecules. Okay, then here the cytosol of bacteria is often differentiated into two distinct areas. The cytosol is in the bacteria, it is divided into two regions. What are that? Means that one is the very a less electron dense nuclear area. Here you see there is a presence of a nuclear area. This is one. And the second one, it is the very dense area, dark region. In the dense cytoplasm occur thousands of particles. Okay, see here, except this nuclear region or nuclear area, we can call this as the dark area. There are two areas. One is the nuclear area. This is the nuclear area. And one more is the dark area. Okay, in the dark area or in the dark region, it is composed of RNA. See, first of all, in the dark region, cytoplasm occurs thousands of particles. How many? Thousands of particles will be there, which is about 25 nanometer in diameter. In this dark area or in this dark region, there is a presence of thousands of particles. Whose diameter is 25 nanometer in diameter. 25 nanometer in diameter. So, what these particles which are present in thousand in number in this dark region or dark area means it is known as the ribosomes. What it is known as? It is known as the ribosomes. Okay, the next here, ribosomes are composed of, what do you mean by this ribosome? These are composed of RNA and protein, and they are the sites of protein synthesis. Ribosomes are composed of two things. What are that? Means RNA and proteins. Okay, as well as ribosomes are also known as the site for protein synthesis. What it is also known as? It is also known as the site for protein synthesis. So what is the here all the ribosome, either it is present in the eukaryotic cell or either it is present in the prokaryotic cell, there is a presence of certain subunits. Okay, so whatever the ribosomes of bacteria are there, it will be always 70 years time. Ribosomes of bacteria, it will be 70 years. How much it is? It will be 70 years time, which consists of two subunits. Okay. See, for example, this is the ribosome. It consists of two subunits. One is the larger one, which is the, see here, this one is the larger subunit as compared to the other. Larger subunit, and another is the smaller subunit. Okay, what is the sedimentation coefficient of this larger subunit and the smaller subunit? Means here it is 50 years. What do you mean by S means that it is the Swedberg unit, which is measuring a sedimentation coefficient of the ribosome. Okay, so it is 50 years and this is the 30 years. Altogether, how much it is? 70 years. Okay, form or many ribosomes will be present in a chain-like structure, or it will be present like this together, and these ribosomes are known as the polysomes. Okay, the next here this is about the polysomes, or else it is also known as the polyribosomes. Except this is about the cytoplasm. Next we will go for the some of the reserve materials which are present in the prokaryotic cell. Next, that is reserve materials. Reserve materials. What are the reserve materials? Reserve means which are responsible for storing some of the substances. Okay, here reserve materials of bacteria are stored in the cytoplasm either as finally dispersed or distinct granules. Okay, here what is the distinct granules means that is the inclusion bodies. What are the reserve materials? First we will talk about the inclusion bodies. Inclusion bodies. Here, what I mean by inclusion bodies means it is one of the reserve material of bacteria in the cytoplasm, stored in the cytoplasm. Reserve material of bacteria stored in the cytoplasm in the form of either as finally dispersed granules or distinct granules which is called as the inclusion bodies. What it is called as? It is called as the inclusion bodies or storage granules. Okay, here in the cytoplasm, here is inside the cytoplasm, there is a presence of some organelles which is stored in the cytoplasm either as finally dispersed or distinct granules you can found over here, and these granules are known as the inclusion bodies or the storage granules. Okay, so this is about the reserve materials. Here then this what does this store? Here, on the basis of that storage, the reserve materials or the inclusion bodies are of three types. How many types of the reserve materials are there? There are three types. First, there is an organic polymers. Okay, organic polymers which either serves as a reserve of carbon. First one you see. First one, there is an organic polymer. This organic polymers will serve as a reserve material for, which serves as a reserves of carbon, which is going to serve or which is going to store the carbon as thus poly beta hydroxybutyric acid as stores of energy. One more, poly beta, second one, poly beta hydroxybutyric acid. Polybeta hydroxybutyric acid will serve as a stores of energy, as it stores the energy. Who is going to store the carbon over here? That is the organic polymer. Who is going to store the energy? Polybeta hydroxybutyric acid. Okay, and these particular inclusion bodies or the reserve materials are known as granulose. Both of these you can call it as the granulose. This is the first type of inclusion body. Secondly, many bacteria contain large reserve of inorganic phosphate. Okay. See, secondly, Many bacteria contain a large large reserve of inorganic phosphate. Inorganic phosphate. Okay, and what is that inorganic phosphate? Means it is one of the highly refractile granule and which is known as the volatile. What it is known as? It is known as the volatile. First is the granulose, second is the volatile. The third type of reserve material is elemental sulfur. This elemental sulfur. And here it is formed of oxidation from hydrogen sulfide. Okay, so this is about the reserve material. Third, we will go for the nucleoids. What is the third structure? Here that it is the nuclei. So let's see about the nucleoid. Next up, it is the nucleoids. Here, in the nucleoids, in bacteria, the nuclear material includes a single circular and double stranded DNA molecule. Here you see, this is the bacterial cell in which this is the nucleus. This nucleus will have a single circular, like this it will be single circular double stranded DNA molecule. Okay, in the nucleus, this is the single circular double stranded DNA molecule, and which is present, which is often called as the bacterial chromosome. What it is called as? It is called as the bacterial chromosome. Okay. Sometimes they will ask for 1 marks. What do you mean by bacterial chromosome? That means the single circular double-stranded DNA molecule which is present in the bacterial nucleus. Okay. So here it is not separated from the cytoplasm because the nuclear genetic material is made in prokaryotic cell. That is the reason it is present in the cytoplasm. Here from the cytoplasm it is not differentiated into what? Into the nucleus, nuclear membrane. Okay. So however the nuclear material is usually concentrated in the specific clear region of the, you can clearly visualize that there is a presence of the nuclear material, and that clear region which is showing, for example, this is the region which is there is presence of what? Nucleus, and that clear region we can call it as the nucleoid. Where a clear region where there is a presence of the genetic material or the nuclear region or the nucleus, that is known as the nucleoid. The next term, a nucleoid will have two parts, that is a nucleoid has no ribosome and nucleolus. Here there will be no nucleolus as well as there will be no ribosome. The bacterial chromosome is permanently attached to the plasma membrane at one point. Okay, it is attached to the plasma membrane like this at one region. Okay, then next thing, here the bacterial chromosome does not contain the histone protein. It does not contain histone protein. Here the DNA is not packed, okay, freely named present in the cytoplasm. So that is the reason there is no histone protein in the DNA. Okay. The next thing here, so this is about what? About the nucleoids. The next topic here, see, all the three classes of RNA will be present in this nucleoid. What are the three classes of RNA? Means there will be presence of three classes of RNA. What are that? Means mRNA, rRNA, and one more, that is tRNA. All these three are present in these nucleoids of bacteria. Okay, these are transformed actively to form a. These are transferred actively to form a enzyme that is known as the RNA polymerase. RNA polymerase of a single RNA. Okay, so this is about what? About the nucleoids of prokaryotic cell. The next is the plasmid, flagella and other structures. These two topics are there of the prokaryotic cell. After that, this is going to complete that. And one more thing, reproduction in bacteria. This topic we will complete in the next class. Okay, thank you.